So thinking back to class the other day, we kind of got through the anatomy of a muscle and the fibers and the myofibrils, which are the contractile organelles. We find the thick and thin filaments arranged in sarcomeres. And as we learn, there's different proteins that make up the thick and thin filaments. In the case of the thick filament, made up of the motor molecule myosin, with its two heads that can swivel, and then the thin filament made up of actin predominantly, but also triple myosin and troponin. Now, if we were to actually go in the same order as the textbook, we would have talked about this stuff way before we talked about neurons. Okay. Well, the reason I talk about the nervous system before I talk about any other system, a couple of different reasons. Well, first of all, it's in charge of everything. Okay, but also, big reason why is because muscle can't contract without first nervous system stuff. Without a nerve signal, a muscle fiber is not going to contract. Okay, and as we know, since we're talking skeletal muscle fibers, we're going to be talking about somatic motor neurons. And if we look here at the illustrations at the bottom, these illustrations look similar to what we learned about previously when we first learned about synapses. What we have there is a synaptic end bulb or a synaptic end knob, whatever you want to call it, where we have vesicles that store neurotransmitters that when the action potential arrives, calcium enters the knob. Those vesicles move towards the cleft, undergo exocytosis, and then the neurotransmitters are released into the cleft. Now, back when we talked about neurons, we kind of focused more on like how neurons can communicate with other neurons. But as you can see here, neurons also can communicate with muscles. Okay, so when stimulated by an action potential, a motor neuron will release acetylcholine, as we've seen that before. Capital A, capital C, lowercase h is that abbreviation into the cleft. Okay, and as I've already stated, remember ACH stored in synaptic vesicles in the synaptic end bulb, or as it's labeled here, the synaptic knob. Okay, and then the acetylcholine comes across the cleft and it binds to receptors which are found on what's called the motor end plate. What is the motor end plate? It's just like a fancy name for the muscle membrane on the other side of the cleft. Okay, so it is just this general area where we're across from the synaptic end knob or end bulb. And as you can see, there are invaginations, these little depressions found within the motor end plate. Okay, and that increases the number of receptors that you're going to have there because there's more space, there's more surface area, so there's more availability of these receptors. Okay, here's an example of one of those receptors. Okay, it receives that acetylcholine like you see here, and then as a response, that opens the channel to allow ions to flow through. Okay, within each motor end plate, there are 30 to 40 million of these acetylcholine receptors binding specifically to acetylcholine. So there's a lot of capability to have acetylcholine connect with these receptors and then allow these channels to open up. We're going to focus in on specifically the opening of what we call ligand-gated sodium channels, which then creates a local or graded potential in the muscle membrane. Okay, now remember, we learned the other day that muscle cells or muscle fibers maintain a resting membrane potential, just like neurons did at that -70 that we previously learned about. And if these, like for example, sodium ions are able to rush in, that's going to change that resting membrane potential. And just like in neurons, we previously learned that they have a threshold. So do muscle fibers, which means that, and we talked about this the other day, muscle fibers can become excited, which means they create action potentials. And that's what we call muscle fiber excitation. So muscle fiber excitation, process in which an action potential in the nerve fiber leads to action potentials in the muscle fiber. Okay, and here's the synaptic knob, so our end bulb of the neuron releasing acetylcholine, binding to a receptor like we just saw on the previous page. sodium comes in, and it's the same two voltage-gated channels that we previously learned about that neurons had. Voltage-gated sodium channels allow sodium to open or to come in. Remember, voltage-gated sodium channels open up when threshold is met. That's going to cause rapid depolarization that then spreads down the length of not the axon, since this is a muscle. Instead, it's down the length of the muscle fiber on the outside, thanks to the sarcolemma. While that's occurring, voltage-gated potassium channels are opening shortly after to allow potassium out to cause repolarization. So it's all about what we've already learned a few weeks ago. Okay? So if a strong enough local potential is created in the muscle membrane due to the flow of sodium ions into the muscle, action potential will be created and propagated across the muscle. So in other words, if we reach threshold, an action potential is created that then spreads from its origin. Now, those figures that you've seen here for the last couple slides are figures I actually have snagged from a different textbook. I really like these different figures because it kind of goes step by step. And so we can kind of go slide by slide. This is one of a few pictures that your textbook has. Your textbook's kind of giving more summary style pictures. So in this case, we show muscle fiber, and all of these structures inside are those myofibrils we talked about the other day. Here's the neuromuscular junction, the point where the neuron and that fat bulb-like structure and the muscle fiber meet. There's the motor end plate. We zoom in on it here. We see all those things that we just talked about, the invagination, so that the number of receptors is increased. We zoom in, we see the acetylcholine coming out, binding to the receptors. Sodium rushes in. If enough sodium rushes in, an action potential is produced. And as we learned, that action potential is going to spread from here throughout the entirety of the sarcolemma. Remember, the sarcolemma is just a fancy term for the plasma membrane, right? But the other day, if I go back to this figure, we learned that we want to take that action potential from the outside of the cell and carry it inward, which is what these T-tubes do. So let's talk about those T-tubes and show an illustration with it occurring. So as the stimulus travels across the sarcolemma, it will travel down into the muscle fiber through its T-tubes. Okay, and that's what we're showing here with step 6. I guess they're using the yellow arrows to denote an action potential going from that neuromuscular junction throughout the sarcolemma and then down into the fiber, thanks to the T-tubes. Now, as we learned the other day, on both sides of a T-tube are what we call terminal cisterns. Terminal cisterna will be the fattened ends of the sarcoplasmic reticulum. Okay, the sarcoplasmic reticulum, as we learned the other day, is where we store and can release calcium. Okay, and the arrival of that action potential triggers what we call calcium release channels to open up, and their name tells you exactly what they do. Okay, they are released out from the SR and into the sarcoplasm, into the cytoplasm of the muscle fiber. Okay, now in a minute we're going to watch a video that goes through all these steps, and it's going to animate it all, and there's going to be a guy narrating it, or a lady, I can't remember, but you're going to see some kind of fancier terms for these release channels. All you need to know them as are calcium release channels. So that's as easy as it gets because it's telling you exactly what they do. Now, all that has led to the point where we can now actually start to talk about the muscle contracting. We haven't really got to that point at all. We're just still in the point of getting the muscle ready to contract. excitation contraction coupling, or if you want to abbreviate that, ECC, refers to the events that link the action potentials on the sarcolemma to the activation of the myofilaments, thereby preparing them to contract. So in other words, it's linking excitation and contraction. That's coupling means like putting them together. Okay, that's just kind of the fancy term that describes this property or this process, I should say, not property. Okay, so it involves, once again, calcium flowing out of the terminal cisternae of the SR. Okay, and then we talked about this the other day, but you're going to get to see it here in the next couple slides. Calcium binds to troponin. Remember, troponin is on the thin filament that helps move the tropomyosin out of the way, opening up the myosin binding sites on the actin. Myosin heads then are able to attach, forming what's called a cross bridge. I used this phrase or term the other day. A cross bridge is just the bond between actin and myosin. So in other words, where the myosin heads are attached to the actin. A myosin heads attached, go through the contraction cycle. In other words, they pull on the actin and will continue to do so. Okay, as long as there's calcium that's allowing for those myosin binding sites on the actin to be exposed. Okay, so the fourth bullet there, myosin heads continue attaching and contracting until all calcium is removed from the cytosol by calcium active transport pumps, which we'll get to here in just a second. So here's a couple more illustrations. We're zooming in at thin and thick filaments. In figure 8, we show, and I actually jumped ahead to this the other day, a couple calcium ions binding to troponin. Now, in this case, the tropomyosin here is covering up those binding sites, but we go from 8 to 9, where now that calcium arriving at the troponin has moved the tropomyosin. Now, I mentioned this the other day, but they're labeled as active sites. It's the same thing as myosin binding sites. So if you want to label that and put in parentheses, it might not be a bad idea, just so you keep your head straight on it. Active sites and myosin binding sites are the same thing. That's just the point where the myosin and the actin can connect. Now, looking at figure 9, we see that both of the heads of that myosin are in a flexed position. In other words, it's like, you know, if you bend your elbow, you flex your elbow, right? Okay. Well, if you want to grab and pull something toward yourself, wouldn't you want to grab with your arm extended? That way, when you pull it, you flex toward yourself. If these myosin heads grabbed a hold of actin and tried to pull, there's nowhere for it to go, and there's no way to pull it because the myosin's already gone through its pulling stage. So what it needs to do is this: extend or cock itself. Okay, and it needs ATP to do so. ATP, as we know, is energy. It breaks it down and hydrolyzes it, and that's going to form ADP and PI. There's where the third phosphate is. Now that's why it's ADP. It is diphosphate. And that's what takes it from that flex position to, they call this like a cocked position. Okay? That way, now the cross bridge can form, where then the myosin head can pull, like it's shown right here. It goes from this position and it pulls itself this direction, and that's what pulls the thin filament with it. Okay? We call that process the power stroke. I'll let you write this stuff out right here. So muscular contraction can be described as a step in which the muscle fiber develops tension and may shorten. Model describing this muscle contraction is called the sliding filament theory, and I talked about this the other day. States that the actin and myosin myofilaments, the thick and the thin, do not become shorter during contraction. Instead, the thin filaments just slide over the thick filaments, so the actin slides over the myosin, causing the fiber to shorten. And we got to see that back with this figure. Now we show the A band never changes length, the thick and the thin filaments don't. Okay, all that's changed is the amount of overlap that exists between the thick and the thin, which affects the I band, or excuse me, the H zone and the I band. Now I used the phrase power stroke. The power stroke is illustrated here with figure 12. Sometimes you'll see this listed as a working stroke. It's the same thing as the power stroke. Okay, this occurs when the myosin head releases that ADP and PI that it had, that cocked it to take it from this position to this position. It still holds on to that ADP and PI, but when it flexes and pulls, it releases that ADP and PI. Okay, and that allows it to flex and pull the thin filament past the thick. And that's why you see the arrows pointing in this direction, because the thin filament's getting pulled in that direction. Okay, now this is only going to shorten the sarcomere very, very minimally. Okay, I'm talking fractions of anything, so it's minuscule. What we need to do is perform this process over and over and over again, which means that we need to, after flexing and pulling, break the myosin head free and then cock it again and then attach it again. And we call that the contraction cycle. Okay, in order for muscle shortening to truly take place, actin and myosin must continually cycle through the contraction cycle. You might see this listed as a cross bridge cycle as well. I call it the contraction cycle. That's what your textbook uses. So that's what we'll go with. Okay. In the resting state, myosin filament is unattached to actin, and ATP is already separated in ADP and PI. Okay, and that's basically where we're at with step one. Okay, and that's what we see here at the top of our figure. Okay, where there's not an attachment, not yet anyway, between the myosin and the actin, so there's no cross bridge. But importantly, what's happened is the myosin heads have gone from the flexed state to the cocked or extended state. Okay, and then as long as calcium's present and the tropomyosin's not in the way, a cross bridge can be formed. Okay, cross bridges, remember, are just the connection between the thick and thin filaments, between the myosin and the actin. Okay, as that occurs, we then can go through step 3, which is the power stroke. Also goes by working stroke, as I've described it before. ADP and PI molecule are released. Myosin head goes through the power stroke or working stroke, causing the sarcomere to shorten. So it goes from this position to being pulled, and in doing so, it yanks and slides the thin filament along with it. Okay. But then, like I said, what we need to do next is to break that cross bridge, which is what we call detachment. Okay, and that requires ATP. Okay, ATP breaks the bond between the actin and myosin, okay, causing it to be released from the thin filament. We see that step 4, so that then that ATP can be used and go through hydrolysis to get it back into the position so that we can do this again. And like I said, this is a cycle, so this is taking place very rapidly. And remember, each myosin head, or each myosin molecule has 2 heads, so they can kind of alternate. Both of them might or might not be in the same stage of the cross-bridge cycle. Okay? This process is going to continue as long as we have 2 things present. Calcium, which once again keeps that tropomyosin out of the way of the myosin binding sites. And then ATP, because as we know, ATP is needed here, and then we release it here as we go through the different steps. Okay, so as long as ATP is present to separate myosin from actin, we can go from step 3 to step 4, and then that allows us to go through the rest of the steps again. Okay. Now, I'm sure you guys have heard of rigor mortis before, right? Well, if you haven't, rigor mortis is what happens after we die or after another animal dies. And it's because of the release of calcium from the sarcoplasmic reticulum, causing this process to keep occurring until all the ATP is utilized. So essentially, calcium's released, there's still ATP left over, so the muscles are actually contracting until the ATP runs out, which means they get stuck in this position right here. where the myosin heads can no longer detach from the actin, and therefore no muscle relaxation can occur. And so that's something that people experience for a few hours after death, okay? And it lasts for about 24 hours. And that's why they call dead bodies stiff, okay? And all the muscles get tight and the body is like stuck in position. It doesn't last forever though, as it says, disappears as proteolytic enzymes, which are specific types of enzymes, digest across bridges. So if you ever wonder where rigor mortis comes from, now you know. So that's muscle contraction. Let's now talk about how we relax the muscle. And we'll kind of start the same way that we started by talking about neurons, which started the whole process of muscle contraction. In order for muscles to relax, we need to stop stimulating them, okay? And muscle relaxation is going to allow the myofilaments within each muscle fiber to return to their resting states. It begins by no longer releasing acetylcholine from the synaptic end bulb, okay? And that happens because there's no longer action potentials arriving there, okay? If there's no more acetylcholine being released, that means no more binding of acetylcholine to those receptors, no more depolarization, no more action potential, no more any of that stuff we just talked about. Okay. Now, to help also eliminate acetylcholine from binding to receptors, we have a special type of enzyme, very long name, acetylcholine esterase, or ACHE for short. Okay. It's going to break down any leftover acetylcholine and remove it from those receptors. Okay. And what takes place is then it's recycled. The acetylcholine can be recycled, so it's not just broken down forever. And as you can see, that's taking place right here. Like I said, in a second, we're going to watch a video that goes through all this stuff. They're going to call it cholinesterase, if I remember correctly, instead of acetylcholinesterase, same thing. Okay. And the figure from your textbook that I'll show you here in just a second actually shows this taking place way before step 15. Okay. And that's because it's not like acetylcholinesterase only appears at this point. Okay. It's actually going to be breaking down acetylcholine even during all that other stuff. It's just, it really starts to work because at this point, you're no longer releasing any more acetylcholine. So then it's, you know, solving the problem for sure. Okay, so with no more action potential in the muscle fiber, we then are going to be triggering the release of calcium from the sarcoplasmic reticulum. And any leftover calcium that's out in the myofibrils and out in the sarcoplasm needs to get put back where it came from. Back in the sarcoplasmic reticulum, back in the SR. And in order to do so, we have some special pumps, what we call the calcium active transport pumps, or as they'll sometimes be labeled, calcium ATPase pumps, which is a good name because ATP is needed, and we should know that by now. Active transport, remember, is where we're going from low to high, and therefore need energy in the form of ATP, although it's not shown in the figure here. Now, inside of the sarcoplasmic reticulum, we have a special type of protein called calsequestrin, which helps bind calcium so that it allows for an even greater amount to be stored. So the moral of the story when it comes to ATP is not only do we need it for a muscle to contract, but we also need it for a muscle to relax. So explains why muscles produce so much heat when they contract. There's a lot of different things going on, a lot of energy being utilized, a lot of heat being produced. And if there's calcium being removed from the sarcoplasm, it means it's being removed from the thin filament. It's been taken away from the troponin, which means that the tropomyosin is going to shift back and cover those binding sites. And therefore, even if that myosin head was in the correct position, which it's not right here, it's not going to be able to bind to the actin anymore. It can't form a cross bridge because those binding sites are covered. Okay. And then the muscle fiber can return to its original resting length due to the recoil of series elastic components. In other words, it's because of that tighten, remember, that allows it to recoil along with just by like contracting your antagonistic muscles. So like if you're looking at the biceps, it would be like the triceps contracting, helps lengthen out the biceps. Okay, so all those figures, and then this one is from your textbook. Like I said, they kind of give more summary style figures in your textbook. We'll start at the top here with number one, a nerve impulse or action potential and a somatic motor neuron triggers release of acetylcholine, binds to receptors on the motor end plates, creating an action potential in the muscle fiber that then spreads throughout the sarcolemma, travels inward thanks to those T2s. Okay. Calcium is then released from the SR through the what we call calcium release channels. Okay. Calcium release channels allow the calcium out that then can travel and find the troponin on the thin filament, bind to tropomyosin, it's going to be bind to troponin, shifts the tropomyosin so that the binding sites are available. Then the myosin heads can connect, go through the contraction process, cycle through that contraction cycle as we just saw a couple slides ago. And that's going to continue as long as we have adequate calcium and adequate ATP. But if that nerve signal stops, all this goes away. We take the calcium back away from the thin filament, put it back into the SR, thanks to those calcium active transport pumps. So like I said, sometimes you'll see them listed as ATPase pumps, same thing. Calcium goes back inside, that requires ATP, as we said. No more cross bridges because the thin filament has the tropomyosin covering the actin. Okay. Now look here at step 3, I kind of skipped it, and like I said, acetylcholine esterase doesn't really just kind of take a foothold in the synaptic cleft at the end. It really truly is there all the time, but it's really, if you're going to continuously produce and release acetylcholine, it's not going to really have a drastic effect until new acetylcholine is not released. Okay, just to summarize here, I've listed off those different steps. I guess I've given 11 of them instead of whatever many it was. A great way to test yourself, it's like what I did and told you guys back when we talked about neurons and action potentials. See if you can write the steps out on a blank piece of paper without cheating and going through all that in your notes to see if you're really ready. Are you going to have to do that on the exam? No, but there will absolutely be questions where you might have to put things in order. I'll maybe give you a step and ask you which happens next. I mean, there's going to be quite a few questions on this, I promise. And then I also will recommend that you can look at these figures and actually explain what's going on. So all these different figures that we talked about, maybe not so much these that are like the summary style pictures, but like these individual ones, be able to look at them and say, okay, yeah, this is acetylcholine binding receptor causing the channel to open up. If I definitely probably will put some of these pictures on the exam. Obviously, I'll take the text away at the bottom, which will give you the answer, but make sure you can identify. I don't do a whole lot of pictures in this class, as you guys know, but there might be a few on this stuff. So now I'm sure your head is spinning right now because this is a lot of information, and I promise you, I kept it pretty simple. There is a lot more intricacies to it. Hopefully watching these videos here will help kind of make some sense where you can kind of visualize instead of just seeing pictures. So
Friends walk up to me and just be like, what the fuck is he about?
okay.
It's not. The process of excitation, contraction, coupling consists of all those processes involved between the time of generation of the action potential on the muscle sarcolemma and the actual contraction. As the action potential reaches the neuromuscular junction, vesicles containing acetylcholine fuse with the membrane of the nerve terminal, and acetylcholine is released into the cleft. Acetylcholine molecules rapidly diffuse across the cleft and bind to the acetylcholine receptors. Binding of acetylcholine to receptors immediately opens ion channels, allowing sodium ions to rush into the muscle fiber and potassium ions to diffuse out. Acetylcholine is then split to acetate and choline by cholinesterase, resulting in inactivation and closure of the channel.
Okay, so there's that acetylcholinesterase. Like I said, they call it cholinesterase, and just illustrating, like I said, that's not just waiting until the end. It's always working, but if we're continually releasing acetylcholine, it's not going to produce a great effect because you're still flooding that acetylcholine into the cleft.
The initial flux of ions due to opening of the channels brings the membrane potential to threshold, causing the voltage-gated sodium channels in the vicinity to open. The resulting action potential is propagated over the entire surface of the muscle and also courses into the T-tubule system. As the action potential encounters DHP receptors in the T-tubules, it causes opening of the ryanodine calcium channels of the lateral sacs.
Okay, like I said, there's a fancier term for what we call them. Those are just the calcium release channels. So you can see why I don't like what they call them in the video. Calcium release channels are a lot more straightforward, right? So each of these little red dots, that's going to be calcium coming out.
Thus allowing calcium ions to diffuse out of the lateral sacs and into the region of the myofibrils. Calcium then binds to troponin, resulting in movement of the tropomyosin-troponin complex away from myosin binding sites on actin. The contraction process continues as long as action potentials continue to reach the neuromuscular junction, thereby keeping the concentration of calcium elevated around the myofibrils. To end the contraction, calcium must be actively transferred by the calcium pump, calcium ATPase, back into the lumen of the lateral sacs.
Calcium active transport pump, same thing.
Utilizes ATP as a source of energy. This process reduces the concentration of calcium in the vicinity of the myofibrils, such that calcium dissociates from troponin. Tropomyosin again covers myosin binding sites on actin, and the muscle relaxes.
All right, so that's one video. I like the next one quite a bit too. So that's why I've got a couple of them here. This one's really good, actually, really, really good. It's a little longer though.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1, cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ATP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3, cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay. There's a lot, I know, but pretty dang amazing. I always tell students to think back, we talked a few weeks ago about like reflexes, how when you like touch something hot and how instantly you pull your hand away from the stove, or when you step on something, or any of the reflexes we talked about, tapping on the patellar tendon and you kick your leg out as a result. And we learned about how crazy complicated action potentials are and how fast those are. And now we understand how complicated a muscle contracting and then relaxing is. So if you don't have some appreciation for how amazing, cool that really is, I don't know what to tell you. It's awesome, cool machines. So sorry, I'm geeking out a little bit, but it's good stuff. All right, like I said the other day, this is going to be a 40-question packet, okay? You can kind of see why. This is kind of, I don't know, I always say this is about the halfway point. So about 20 of the questions will come from, the beginning of this lecture to this point, which means about the other half will come from here on the rest of the way. And this is where we get to talk about some different things related to muscles and how they work. We're going to talk a lot about force that muscles can produce. Force, or like how strong a muscle contracts. The word tension, you'll see several times, it's the same thing as force. So in other words, length tension relationship, you basically could say length force relationship, which is going to state that for skeletal muscle, it indicates how the forcefulness, there's that word forceful, forcefulness of muscle contraction depends on the length of the sarcomeres within a muscle before the contraction actually begins. So in other words, force is dependent upon the number of cross bridges formed by the actin and the myosin molecules, and it all relates to what position the muscle was in before it contracts. Okay, we've got 5 different things listed here, and there are 5 different things listed here, and there's 5 different points here on this graph. This is actually the figure from the scientific study that first described this process. Actually, it was really cool. This experiment, what they did was they took a muscle out of a frog, and so the frog was no longer alive, and they took the muscle out, and they put the frog's muscle in a different position. So as far as length, it was short, it was a little bit stretched, it was stretched and stretched, and then finally it was super stretched. And they hooked it to a machine that could zap it with like an artificial action potential, and they measured how forcefully it contracted. Okay. So at point 1, which is right here, I know it's kind of blurry, this is number 1, so is this, okay? What we're measuring here is the length of the sarcomere. So in other words, how long the muscle is. Here we're measuring how much force. So at the point where the muscle is at its shortest, can the muscle shorten anymore? No, okay? So can any force be produced? No, okay? So in number 1, no force can be developed because the Z discs, and that's what we see down here, are so close together, and thus the myosin and actin are smashed together, and therefore no force can be produced. Whereas we go to step 2, point 2, and we lengthen the sarcomere just a little bit, look what happens to the amount of force that can be produced. Okay, we stretch the sarcomere to the point where now we can actually shorten it when it contracts, okay, and thus force can be produced. Okay, point 3, myosin and actin are spaced properly to reach what we call peak tension, and which is where we're going to max out. Okay, at points 3 and 4, we hit what we call L naught, which is where it's at max force or max tension. And that has everything to do with the fact that it's at a perfect length, which means we have the perfect amount of cross bridges. Okay, the more cross bridges you have, the more force. Okay, you think about it that way. The more cross bridges you have, the more force you produce. Okay, and points 3 and 4 are where we're at the perfect stretch, the perfect overlap between the thick and thin filaments, and then we can have the perfect number of cross bridges. Okay. Point 5, look what happened to the sarcomere here. Here's the thick filament, here's the thin filament. They're no longer overlapping. Is there any way for a cross bridge to be formed there? If there's no cross bridge, no force can be produced. Okay. So in point 5, myosin and actin are overstretched to the point where no cross bridges can be formed between the two filaments. Now, are you probably ever going to experience a point where you're like this inside your body when your joints and your muscles are put together? No, absolutely not. This was, like I said, taken in a study where the frog's muscle had been removed. Okay, the way that our joints and our muscles are positioned, we're built to be somewhere in this neighborhood. Okay, but we can, lift things and do different motions with our joints in different position, where our muscle might be more stretched out or it might be more shortened, which is going to greatly reduce the amount of force that you can produce. Okay. Make sense? Okay, it's kind of a crazy concept, but a really cool one at the same time. Another very important thing to talk about when it comes to muscles, and we've been talking a lot about it already today, is the fact that it needs ATP. A huge amount of ATP is needed when muscles are contracting. It's needed to power the contraction cycle. It's also needed to relax the muscle and pump that calcium back into the sarcoplasmic reticulum. It's also very involved for other metabolic reactions involved in muscle contractions that we didn't get into. We haven't really got into great detail about how we produce muscle or how we produce ATP in this class. I kind of introduced it back in Unit 1. We talked about the terms aerobic versus anaerobic, with oxygen, without oxygen. Okay, you see some of those words again here today and on this slide. Okay, there's 3 ways that muscle fibers produce ATP, and we're just going to really briefly talk about this stuff. Like I said, we haven't talked too much about it. We'll get into all about respiration and mitochondria and all that wonderful stuff later in the semester. So this is going to be a brief intro into it. Okay, when our muscle fibers are in a resting state, like the muscles in your legs are probably right now, if you're not doing much sitting around, we are able to produce more ATP than they need for that resting metabolism. So we can kind of stockpile it. And we do so by taking that excess ATP and we synthesize what's called creatine phosphate, which is exactly what its name sounds like. It's creatine with phosphate attached to it. The phosphate, it's an energy-rich molecule only found in muscle fibers, so it's not like you're going to find this in your brain. Okay. There's an enzyme called creatine kinase that takes the 3rd phosphate from ATP and transfers it to the creatine molecule. Okay. And what you're left with as a result is that ADP with the 3rd phosphate no longer there. The creatine has that phosphate. That way, when the muscle begins contracting and it needs that ATP, you can just give the phosphate right back to that ADP and form the ATP. Okay. This process is anaerobic, which we've seen before. Once again, means no oxygen is necessary for this process to occur. We utilize this type of energy substrate when we need to do something really vigorous and really short in duration. Okay? It can provide enough energy for muscles to contract maximally for about 15 seconds. So like going from outside, whether you're here in Atchison or Wamego, and I just all of a sudden said, okay, run as fast as you can down the street. Going from that resting state to that maximal contraction, your ATP needs are going to increase dramatically. Thankfully, we don't have to go through anything too complex to make that ATP. But then again, this is going to be just really short term because only 15 seconds. I would hope everybody could run for longer than 15 seconds. Examples of types of athletic events that we will be utilizing predominantly creatine phosphate, like a 100-meter dash, like running just a little longer than the length of a football field, or a set of lifting weights. Now, you've probably heard the term creatine before, right? Anybody heard that term before? Aspen shaking her head. Yes, when have you heard of creatine? Yeah, it's a supplement, right? And, well, guess what? People take creatine. They have more of this, which means they can have more of this, which means they can produce more of this. Who commonly uses creatine? Bodybuilders. And like I said, a set of lifting weights. Doesn't technically make you stronger, doesn't make your muscles get bigger, but it gives you more energy so that maybe you can do an extra lift or two, and that's what makes you stronger.
So it's like an energy drinks and
Kind of. I mean, energy drinks are just caffeine, which we're going to get into all that stuff. I'm kind of getting ahead of myself, but it just gives you more energy to do the extra rep or two. Now, once we run out of the creatine phosphate in our muscle, we got to turn somewhere, which means we're going to use the next method, ATP from anaerobic cell respiration, which right away should tell you that oxygen is not involved here either. Okay, since the word anaerobic is there at the top. This is once again what occurs after a muscle has depleted supply of creatine phosphate. This process involves the movement of glucose in the contracting muscle fibers, where it is broken down in a series of 10 reactions, which we call glycolysis. Now, in order for this to occur, we actually have to use a little bit of energy in the form of ATP, 2 of them, to form a total of 4. Think of this like if you go to the casino and you put 2 quarters in and you pull a lever and 4 quarters come out. How much money did you win? Not 4, you want 2, right? Okay. So that's why you see 2 ATP over here. It's a net of 2 instead of a total of 4, because you had to use 2 to get to that process. Okay. Now, what you form as a result of this is 2 molecules of what's called pyruvic acid. Depending on the availability of oxygen in the muscle fiber, you can either use that pyruvic acid and throw it into the mitochondria, because this takes place outside the mitochondria. That process is what we would prefer to do. And that's where we're going to end up using oxygen and making a lot more ATP. But if we don't have enough oxygen in our muscle fibers, and so we'd say we have anaerobic conditions, we're going to take that pyruvic acid and convert it into lactic acid. Now, lactic acid, I imagine you guys have probably heard of before. It kind of gets a bad rap. People like to blame a lot of things on lactic acid. You guys ever heard of lactic acid before? Aspen's shaking her head, yes. What have you ever heard about lactic acid?
yeah, I've heard of that. I don't know what that is.
People like to say that it causes cramps and muscle soreness and that kind of stuff. Nothing to do with that. It does, I mean, it is an acid, so it is going to mess your body up if you have a lot of it. Okay. But our liver takes a good job, or does a good job typically of buffering out and keeping you fine. But it can reduce your pH. It does produce fatigue, though. It does make you more tired. It does make your muscles less effective. It doesn't cause you to be sore, though. people like to think, oh man, they wake up the next day, I got all this lactic acid in my muscles. They don't know what they're talking about. Now, in the case of this form of ATP production, it's going to provide enough energy for about 30 to 40 seconds of maximal muscle activity. So not quite a minute, about half a minute. A good example of a type of activity, physical activity that would use this as a predominant source will be running a 400-meter race. If you know what a 400-meter is, that's one trip around the track. Obviously, I don't even know if anybody can run it in 40 seconds. That's extremely fast. But most of that race, you'd be utilizing this as your primary ATP source. But most athletic events, most times when you're being physically active, you go longer than 30 to 40 seconds. How cool would it be though if we got to work out for 30 seconds and that'd be enough for the day? It would be awesome. Which means we have to do something else. Okay. ATP from aerobic cell respiration is the most complex of the processes. And since it's aerobic, it's oxygen requiring. And this is all taking place inside the mitochondria. Once again, only occurs if sufficient oxygen is present. And this is where we take that pyruvic acid that we formed in glycolysis right here. If we didn't form lactic acid, instead, we take that pyruvic acid, bring it into the mitochondria. We then go through oxidation and we produce a huge amount of ATP, a lot more than what we did earlier, 30 to 32. And if you add them all up, you get a total of 36. That's from glucose. Okay. You can also perform this process using amino acids from the breakdown of proteins, or you can use fatty acids from your fat in your body, which actually produce a heck of a lot more ATP, over 100. So that's very significant. And that's why this type of ATP production can provide us with enough energy to go for a long period of time. Okay. In activities longer than 10 minutes, about 90% of the ATP we use is provided in this mean. If you're going even longer than that, it's almost 100%. Like in a marathon where you're running for hours, triathlon, that kind of stuff, it's nearly 100%. And like I said, we're going to get into all this stuff much later in the semester when we, get into cell metabolism and that wonderful stuff. That's our second to last unit though, so we got a few weeks before we get to that point. Not that I'm going to ask you to know this. I snagged this actually from my exercise physiology textbook that I teach with once in a while. It shows you kind of percentages aerobic versus anaerobic on the left there as far as time goes, versus on the right gives you examples of different athletic events. Looking at time, that's kind of what we focused on. We said 1 to 3 seconds, 1 to 10 seconds, you're predominantly being anaerobic. As you get farther and farther along at 2 minutes, that's about the point where it's going to shift gears from more anaerobic to aerobic. By the time you get to 10 minutes, it's about 90-10. And then, as you get into the long distance type activities like marathons, it's almost 100%, if not completely 100% aerobic and 0% anaerobic. I have a lot of athletes on campus. I've got like several volleyball players and football players and baseball players and that kind of stuff. So it's kind of cool. Sometimes they're interested in seeing where their sport falls in. Some sports it's kind of unique where you got different positions, like or not baseball, but like soccer is a good example where like you have a goalie, they're staying put and by the goal for most of the game, while you got like midfielders that are running all over the place. So this is kind of like an average, but as you can see, where different sports and athletic events fall in. It's kind of cool. Like I said, you don't really need to worry about any of that stuff, but still food for thought. All right, I've been talking a little bit about fatigue. Let's talk a little bit more about it. Muscular fatigue is defined as the inability of a muscle to maintain force of contraction after prolonged activity. Now, there's actually two different types of fatigue, central and peripheral. Central is that feeling of tiredness and the desire to cease activity prior to actually feeling true muscle fatigue. Okay. In other words, like, when you're jogging or you're walking around and you're feeling tired and you can go farther, you just don't really want to. And as they say, this is kind of what separates the haves from the have-nots or whatever. We call it central fatigue probably because it's caused by changes associated with the central nervous system. And although they're not exactly sure what the exact mechanism is, it's a thought to be as like a protective thing that keeps your body from continuing to go where you might experience muscle damage. And peripheral fatigue involves factors located after the neuromuscular junction, NMJ, short for neuromuscular junction. So that's where the neurons and the muscles are meeting. In other words, like where that synaptic end bulb releases acetylcholine we saw earlier today. Okay. This is what muscular fatigue, this is what actually causes it. Okay. At least most evidence points to this. Okay. Precise mechanisms causing it are still not clear. There are several thoughts that, or there are several factors that are thought to contribute. Okay. An inadequate release of calcium from the SR, which would result in a decline of calcium concentration in the sarcoplasm. And as we know, calcium is absolutely essential for cross-bridge formation. And if you don't have cross bridges, you can't produce force. Therefore, the muscle's diminished in its ability to maintain force. If you deplete your creatine phosphate, as we just saw, that's an energy substrate. Insufficient oxygen, which means you can't produce enough ATP aerobically. If you deplete your glycogen and other nutrients, you would call that hitting the wall. Anybody ever heard that phrase before? It's basically like when your car runs out of gas. If your car's out of gas, can it go any farther? Exactly, your car stops. I'll show you a video here in a second. It's an extreme example of somebody hitting the wall. Actually, it's two ladies and an Iron Man, which is one of the.
Country runners that keep running, they get to the end and just fall down. Like, I'm so done.
Yep, that's exactly what it is. Now, keep in mind, you're not going to hit the wall like, if you do like weekend 5Ks and that kind of stuff. Hitting the wall, that's, where you've been exercising for hours and hours and hours. As I mentioned earlier, lactic acid gets blamed for a lot of things, but it is a definite contributor to fatigue. Failure of action potentials in the motor neuron to release enough acetylcholine. So if you're not getting enough acetylcholine, you can excite the muscle and then it can't contract. And then high hydrogen ion concentrations, which generally occurs with heavy exercise, and that relates to lactic acid because lactic acid is an acid and that's what produces high hydrogen ion concentrations. All right, let's watch this video. It's kind of you might laugh. It's pretty wild.
This is the world's first reusable cotton swab.
Not this
You can
video.
remove up to 10 times more earwax.
That's gross.
Sean Welch is in a desperate spot. Her body has run out of fuel. No calories to burn.
Okay, so if you're not familiar with what an Ironman is, it's like one of the most insane things that people do. It's got a biking, swimming component, and then they top it off by running a marathon.
Yeah.
So it's like over 100 miles of physical activity all in one day. To me, sounds like the worst thing. I don't even like to drive an hour or 100 miles, but these people are incredible athletes and extremely highly motivated. So let's watch.
Systems shutting down. The legs are there, you just can't feel them. The eyes still see, but through a gauzy veil of delirium. At this point, Sean Welch is aware of two things. The finish line is so close, and someone is so close behind. After more than 140 miles,
140, yeah.
it's come down to less than 100 feet to that damn line. Just yards behind, her stalker appears. It's Wendy Ingram. Their eyes roll.
All right, I'm just gonna stop it there because they go on for another minute, just kind of talking about nonsense. That was even for first place. It was like for third or fourth.
Wow.
Pretty wild. I mean, those poor ladies, highly, highly motivated just to even sign up for that type of activity, but to go like that, that's extreme. Like I said, it takes a long distance type activity where you're gonna experience something hitting the wall, not like going for a 5K or something. If any of you guys watched the TV show The Office, there's an episode where they're doing a 5K, and right before the 5K, main character is eating fettuccine alfredo, trying to prevent running a 5K. That's 3 miles. That's not gonna get you anywhere. What that person was doing, Michael Scott, if you watch that show, it's a fantastic show. He was taking and utilizing what's called an ergogenic aid. What are ergogenic aids? Well, those are defined as work-producing substances, a phenomena believed to increase performance. So in other words, it's just something that you might utilize because it might help you do better. There are endless examples of ergogenic aids. Could be as simple as just like listening to music while you're lifting weights. A lot of people like to do that. Stretching before a run or doing a gymnastics routine. Okay, pretty common stuff, right? Well, I chose just a few of them. I already kind of talked a little bit about creatine. Creatine monohydrate, as it's more fancily known, been shown to increase your phosphocreatine levels in the muscle. Phosphocreatine, creatine phosphate, that's the same thing. It's just where the words are kind of split one word from the other, vice versa. Okay, if you have more phosphocreatine or creatine phosphate in the muscle, you can produce more ATP. Therefore, your ability to maintain force and power is enhanced. Like I said, creatine monohydrate used in like bodybuilders. I think Aspen was one that said that. Hey, if those ladies took creatine monohydrate, it would not do them any good because that's not the type of energy you're using to run 140 miles, bike that far. And what they would want to do, like I was talking about with the show of The Office, they would want to carbo load. Carbohydrate loading has been shown to increase the glycogen levels in skeletal muscle in the liver. And therefore, when you're exercising, you can break down that glycogen, release the glucose, and use that glucose to make ATP. Basically, just think of that like going on a road trip. Before you go on a road trip, what do you need to do with your car? Put gas in it. Fill up the gas tank all the way that you can, right? More gas you have, the farther you can go. That's what carbo loading does. Okay. Not beneficial if you're, lifting weights because you're not using glycogen to lift weights. You're using your creatine monohydrate or your creatine phosphate. But it is beneficial if you're doing long distance type activities like those ladies were. And then caffeine, one of everybody's favorite things, not everybody, but a lot of people, myself included. Okay, not only is it good for keeping you awake and waking you up in the morning, but it also can be a pretty darn good ergogenic aid in physical activity. It may improve performance of the muscle or nervous system, and that could even include enhanced delivery of fuel to muscle. It can elevate your blood sugar and increase the utilization of fats, and it can actually make you feel less tired, which is common sense. Like when you're exercising, you don't feel as physically worn out. We call this a lower rate of perceived exertion, RPE. Rate of perceived exertion means how hard does it feel like you're exercising. And if you're exercising at your hardest, you'd say it's like at a 10, out of 1 out of 10, right? And well, if you drink a little coffee or chug down a Red Bull, maybe it goes from a 10 to an 8. And therefore, if you don't feel as tired, you can go a little bit farther. Therefore, your performance is enhanced, right? Okay. Even though each skeletal muscle fiber has only a singular neuromuscular junction, the axon of a somatic motor neuron branches out and forms neuromuscular junctions with many different muscle fibers. So we've been talking a lot about the neuromuscular junction here today. We can see an illustration on the far right, similar to what we saw earlier today, with a synaptic end bulb. Across from it is the motor end plate, where those acetylcholine molecules bind to receptors, trigger all that stuff we talked about. And what you see here is found at each and every one of these little big bulbs at the end of a motor neuron. Okay. And as you can see, we've got a green motor neuron and a purple motor neuron. Okay. And they branch to feed not just one muscle fiber, but in some cases, a heck of a lot. Okay. We call this a motor unit. It's one somatic motor neuron and all the muscle fibers it innervates. So in this case, this motor unit would be this green neuron plus this, and this muscle fiber. Okay, the purple motor neuron innervates these two muscle fibers. So that's the motor unit. Remember, when an action potential comes down the axon, it goes to every single one of these about the same time, which means all three of these in this case, or these two in this case, all those muscle fibers are going to be going through the same stages together. They're going to be contracting in unison. Okay. It can be as few as 20 muscle fibers. That's not very many. It can be as many as 1,000 muscle fibers per nerve. Okay. And it's dependent upon the type of control you need. Okay. If you want to contract really forcefully, the more fibers you produce, or the more fibers you excite, the stronger the contraction is. Whereas if you want to be really precise in your motion, what we would call fine motor or muscular control, we'd use a smaller motor unit, which means that we're going to use the least amount of muscle fibers as possible. Okay. Where would we want to do like fine motor control? Your fingers in your hands. So like when you're writing and drawing, I don't have any of that good stuff but like think about like when we're kids. My kid's in kindergarten now, his handwriting is atrocious, but it's gotten better since the beginning of the school year. Okay. He's developing better fine motor control. Okay. The actual finest motor control that we have is the 6 muscles that move our eyes. Think about how we can move our eyes and how we can pinpoint things. And we can precisely locate by putting our eyes specifically where we want. And that involves the contraction of 6 different muscles. Some of them make you look up, some of them make you look down and roll your eyes and do different things. Okay. When we want to contract really forcefully, we're going to use what we would call strength motor or muscular control. This produces a bulk contraction because you're using as many as 1,000 muscle fibers at the same time. And we're going to find this type of motor unit in your big, powerful type muscles. So like your legs, your back, your butt muscles, that kind of stuff. And thus, very, very strong. All right, a single nerve impulse in a somatic motor neuron elicits a single muscle action potential in all the skeletal muscle fibers with which it synapses with. An action potential always is going to have the same size in a given neuron or muscle fiber, and we've seen that before. Okay, it depolarizes the threshold, then we depolarize somewhere to positive 30, 35, and then we repolarize back to resting membrane potential. It doesn't matter what that original stimulus and how far or how strong of a strength it has. Okay. Now, in the case of muscle fibers, the force that those muscles contract at can vary. In other words, muscle fibers, when stimulated by an action potential or multiple action potentials, can increase their force of contraction. And we see that here in a couple slides, okay, where if we're measuring force of contraction, okay, one action potential produces a little bit of a force, okay, but if we hit it a couple times, then we increase the force, okay. Remember how we talked about summation? We're going to get to that point here in just a second, okay. The total force or tension that a single muscle fiber can produce depends mainly on the rate at which the nerve impulses arrive at the neuromuscular junction. Okay, we call this number of impulses per second frequency of stimulation. So what that means is force is dependent upon how quickly we're stimulating the muscle fiber. Okay. A twitch contraction is actually what we show here on the next page, is a brief contraction of all muscle fibers in a motor unit in response to one singular action potential. Okay, we don't do this naturally. Okay, we don't just send one action potential, muscle contracts, and then it relaxes. It's only able to be done with lab software, okay, and by directly electrically stimulating the motor neuron or its muscle fibers, and it involves 4 periods. Okay, although you can only see 3 of them labeled here, the 4th period, refractory period, is an important thing that I'll talk about after I get time with the other 3. Let's first talk about the latent period. The word latent we've seen before, or we'll definitely see more later in the semester. Think latent as in like delay. Okay. What we're seeing in this graph is down here at the bottom, we're measuring everything in milliseconds, and in the y-axis, we're measuring tension or force. This point right here where the arrow is pointing is showing you when we zap the muscle. Like I said, we can only produce a twitch in a lab. So when the lab software or the piece of equipment stimulates the muscle, in other words, this is where an action potential is created. Okay. But right away, and it's not very long, but there's a period, just a few milliseconds before we do anything to produce tension. Okay. And so this is where all those steps that led to a muscle to contract are taking place. So in other words, it's where the action potential sweeps over the sarcolemma, calcium's being released from the SR. Okay. We haven't quite got to the point where the myosin has attached to the actin and pulled on them. That's what happens next during the contraction period. Okay. Calcium binds to troponin, myosin binding sites exposed, cross bridges form. And then we go through the contraction process where the myosin pulls on the thin filament and peak tension is developed. And relaxation occurs after that when we transport the calcium back into the SR, the myosin sites become recovered by the tropomyosin, and the myosin heads can no longer attach to the actin. And now we've seen this word or this phrase term refractory before, back when we learned about neurons and action potentials. We talked about how, like, if you jump in the air, you can't take a second jump until you hit the ground, right? Okay. Well, the refractory period in a muscle refers to that when a muscle fiber is contracting, it cannot respond to another action potential. So it's kind of the same thing. If it's going through an action potential, it can't go through another one at the same time that's already going through the first one. It has to wait. Okay. Okay, so thinking back again to that same lecture on action potentials and all this wonderful stuff we've kind of referred back to today, we talked about a process called summation. Okay, remember how we talked about if you hit a neuron a second time with the stimulus, it can add those depolarizations together. Okay, well, the same thing can happen to muscle fibers with regards to the amount of tension that they produce. Okay, so like that of nerve fibers, when a second stimulus occurs in skeletal muscle after the refractory period of the first stimulus is over, but before the fiber has relaxed. So in other words, after we've depolarized and contracted, but before we go back to resting tension, we hit it with another action potential. We summate, we add force to it. Okay, so it's progressively going to get stronger. That way, if we continue to hit it with more and more and more, we progressively grow in strength and grow in force of contraction. That's what wave summation is. Additive effects of contractions of different muscle fibers causing a second contraction being greater than the first. And now, I'm sure you've heard of tetanus before, right? In fact, we talked about getting a tetanus shot and why we need to do that, right? Okay. Well, tetany in your muscles is where those muscles are continually contracting. There's two different forms of tetanus, unfused and fused, or incomplete and complete. Incomplete or unfused occurs when the electrical stimulation occurs so rapidly that the relaxation time between twitches becomes shorter and shorter. And that's what we're showing here. Okay, the action potential are pretty close together, but not nearly as close together as here. So what we see is we contract, we start to relax, but then we zap it again, we contract, start to relax, we contract, start to relax, and we're growing in strength of contraction. Okay. Complete or fused tetany or tetanus occurs when the stimulation allows for no relaxation. So we're not dipping back off. Instead, what's going to occur, since we're not dipping back off, is that there's no relaxation periods between the twitches, which allows for the contraction to be smooth and sustained. So it's not like it's kind of herky jerky. Kind of. Okay. if you were to fail to get a tetanus shot, like if you step on a rusty nail, this is what's going to happen to the muscle. It's going to contract and it's going to do so, and you can't stop it from doing it. You can't relax the muscle. Okay. Have you guys ever like been hooked up to like a stim machine? And you can trigger this and muscle if you crank the voltage up. Okay, what's going to happen if you put that like you put the pads? You know what a stim machine is? It's like it's an electrical stim machine that excites your muscle like your neurons can.
Like
And
today's-
it's kind of like being tased. Absolutely, you put those pads right on the muscle or right on the skin outside the muscle, obviously, and you can control how much of electrical signal goes into the muscle. Okay, you can actually cause your muscles to contract, kind of like what you see here. If you crank it pretty far, this is what's going to happen to your muscle, and it's going to hurt. And when I was in school, we worked with this kind of stuff, and so we did labs where we kind of practice, and we all had to take a turn, doing it where you cranked it all the way up, and it does not feel good. Now, why do people do that? Well, actually, stim machines are really good as far as like physical therapy and rehab and that kind of stuff. So if you ever go to like physical therapy, if you want to become a physical therapist, you probably get to use those a lot. It's kind of a different thing for sure.
I've been tased and I'd rather be safe.
I've never been tased, and I'm pretty proud of that fact.
I wasn't arrested, I was a cop. So.
I'm not even saying that. I just
had
I
to
would
do
not
it,
ever want to be
but-
tased. That just sounds miserable.
It was. Yeah, it will never touch my leg again. She did my back. I don't like that at all. Like my arm kept twitching afterwards, and I was like, oh my god, take it
Yeah, it's
off.
weird. All right, so we've been talking a lot about motor units, and remember a motor unit, if I go back a few slides, is one motor neuron and all the fibers innervated by that neuron and how they all contract at the same time. Well, when we do different things, whether that's something as simple as taking your pen or your pencil and contracting those muscles to take your notes, or getting up and walking across the room, and we've kind of talked about this before, but we don't utilize 100% physical effort. And think how crazy and weird that would be if you were taking notes with as hard as you could contract the forearm muscles. Okay, you rip holes in your pages, and you break your pencil, and you'd be exhausted because your muscles would be worn out instantly. Okay, so we don't have to recruit every single muscle fiber when we do simple, easy tasks. Okay, but when we progressively need to produce more force, we can recruit more and more motor units. And that's what motor unit recruitment is. Its name is 100% straightforward. The process in which the number of motor units increases is called motor unit recruitment. Okay. Typically, different motor units of an entire muscle are not stimulated, contract in unison. Okay. In other words, while some motor units are contracting, others are relaxed. Okay. This pattern of activity will help delay fatigue in our muscles, allowing for contraction of a whole muscle to be sustained for longer periods of time. Okay. If we are needing to do something that requires very little effort, we're going to recruit our weakest muscle or motor units first. Okay. If we need to progressively use more, what we're going to do is recruit the larger motor units. We go from small to medium, and then finally, we need to produce a huge amount of tension. We're going to recruit the biggest ones and the strongest ones last. And we save the big ones for last. Okay. And this recruitment of our motor units is another one of those factors that helps us produce smooth movements rather than a series of jerks. And so it kind of coincides with what we were just talking about with regards to wave summation. Okay, muscle tone is something that we've talked about a variety of times. Thinking back a few weeks, back when we talked about the brain, which we actually talked about this morning in anatomy, we learned that muscle tone is a very small or weak contraction in our muscles, even during their most rested states. Okay, so even at rest, skeletal muscles exhibit this muscle tone, a small amount of tension in muscles due to weak involuntary contractions of its motor units. It's established by neurons in the brain. Okay, and specifically, we learned about that earlier in the semester. It also involves spinal cord signals coming out, exciting the muscle motor neurons, releasing small amounts of acetylcholine. Okay. However, if the motor neurons that serve a skeletal muscle are damaged or cut, that can produce a muscle that's flaccid, which is where the muscle is in a state of limpness in which the muscle tone is lost. So in other words, there is no way for the muscle to contract because there's no way to stimulate it, at least not naturally. You could hook it up to a stim machine, but still, what's the point in that? Okay. To sustain muscle tone, small groups of motor units are alternatively active and inactive. Now there's two types of muscle contractions that we can perform: isotonic and isometric. When we think of like contracting our muscles, we're probably going to think of isotonic, which is where a body, limb, or a joint actually performs motion. And in an isotonic contraction, the tension or the force of contraction is developed and remains constant while the muscle, and the key here is that it changes its length. So in other words, the muscle can lengthen and or shorten. Okay, this is what we do when we do most of our motions that we think of when we talk about skeletal muscle contraction: picking things up, jumping, throwing a ball. A example, like they're showing, they're picking a book up off the table. There's two different phases to a singular isotonic contraction: the concentric and the eccentric. Okay, the concentric, you can see there in figure A, and this would be for the biceps muscle, is where the muscle is shortening. Okay, and it's pulling a structure such as tendon, producing movement, which reduces the angle of the joint. In this case, the elbow is being reduced as far as the angle goes. Okay, the eccentric phase is where the muscle is lengthening during a contraction. So in other words, where the elbow is straightening back out, there's still tension in that biceps muscle, but the key is the muscle is lengthening versus when it's shortening. Okay, one of the biggest mistakes I would say that people that just begin lifting weights for like the first time and aren't very experienced is, like, you know, if you think about like doing a bicep curl like the guys kind of doing there, they lift it up and then they drop it, and they let their arm use the gravity to straighten back out, and they lift and they drop. They're basically ignoring a very important phase, the eccentric phase, okay, because they're letting gravity help lengthen their muscle instead of allowing the force to continue to develop as they straighten the joint back out. Okay. Like I said, most of what we would think about as far as types of muscle contractions, we'd be thinking about isotonic. But we also have what's called isometric contractions, which is where we generate force, but it's not enough force for the object to be moved. Therefore, the muscle does not change its length. So like holding a book steady using an outstretched arm, or I always tell students, think of, if you've never done one of these, do one at home, a wall sit. So know what a wall sit is?
Yeah.
Yeah, get up against the wall, get into a squat, or like you're sitting in an imaginary chair, and just sit there for 10, 30, 40, 50 seconds. And you'll know after a few seconds, or like a plank. Okay. We think about like athletics, most sports utilize isotonic contractions, like throwing a baseball, jumping and spiking a volleyball, shooting a basketball. Joints and motion is occurring. Okay, but there are sports that utilize a lot of isometric contractions. Think of any? Gymnastics is a good one. You guys ever seen like when they are doing rings and they're holding their arms straight out and they're holding their body in a position very amazingly, that's a perfect example of an absolutely insane isometric contraction that I couldn't even dream about being able to do. And so gymnastics is a good one. They use a lot of isometric contractions, like I was talking physical therapy. I had to do a lot of wall sits after I tore a ligament at my knee. The great thing about isometric contractions, you're working muscles without working a joint. So like, if you don't want the knee to bend too much and go straight bent, where you're, like when you're running and jumping and that kind of stuff, and you can build back muscle by doing isometric contractions without altering the position of the joint a whole bunch. So. Question? Everybody good there? All right, I'm going to stop here. This is a perfect spot to call it a day. And then we will be able to start from here on Tuesday.